Hi, everybody. Welcome to another Chats with Chaudhry. Today, I'm delighted to be joined by Nagarjuna Nagaraj, who is the Applications Development Manager at Bruker Daltonics. He's nodding his head because I got his name perfect there. So we are going to be talking about some of the new product developments that were announced last week by Bruker at their Bruker Exceed symposium that they had, in which I was lucky enough to attend. So, Nagar, how are you, my friend? Lovely to see
I'm
you.
doing really good. Thanks a lot, and thanks for the opportunity. Yeah,
Great. And where are you calling in from today, Nagar?
I'm living in Munich, so but I'm working in Bremen, in Germany, in Bruker's facility. So I'm kind of shuttling between Munich and Bremen. So I'm calling from home.
Fantastic. Well, look, before we start talking about some of the new developments that Bruker Daltonics announced last week, could you give people not familiar a very quick overview of Bruker Daltonics and also your own background?
Sure, yeah. So Bruker, I think many people know it's kind of well known for its analytical instrumentation. It's including mass specs, NMR, IR, microscopy, and many other instruments. And Bruker Daltonics is the mass spec division of the company. And as a way of introduction to me, so I did my PhD and postdoc in the Matthias Mann's lab. And then I was running the core facility in the Max Planck Institute for Biochemistry in Munich. And this was for about 6 years. And then I joined Bruker about almost 2 years ago. And I'm working in this proteomics applications development team. So we are really developing new proteomics methods and help the customers get better data from their instruments.
Perfect. So let's talk about proteomics, etc. So, what developments have you seen over the last 12 to 18 months in single cell research?
Single cell, I think it is actually an exciting area of research, right? So, which is not only trying to answer some fascinating questions, but also kind of driving these technological innovations and developments. So, and also in single cells, it's been a lot of genomics and transcriptomics, which who take the primary share of that. And we could even see publications with more than close to 1 million cells being studied in a single cell manner, right? So this is impressive. And but actually, it is also important to say that proteomics, which studies the proteins and the proteins modifications, which we call epiproteomics, and other omic technologies like metabolomics and lipidomics, they actually will we really need them together so that we can have a complete systems biology level of understanding of a single cell. And therefore, I think the mass spec is truly key here, right? But proteomics of single cells is kind of relatively a small part, and it is actually really getting active in the last 3-4 years, right? And we also have now single cell, the dedicated single cell proteomics conferences, which is really good for the community. But as a proteomics community, we really need to step up and kind of begin complementing genomics and transcriptomics. And so we start to see some really nice publications, but I think this is only the beginning, and it's an exciting road ahead. Yeah.
Fantastic. So let's start talking about some of the launches which were announced last week. So the first one, obviously, recently launched is the timsTOF SCP. So why is that revolutionary for cell biology and cancer research?
Yeah, the timsTOF SCP is an exciting launch, and actually, the timsTOF SCP is the first dedicated machine that's kind of launched for single cell proteomics with ultimate sensitivity, right? And this instrument is a result of completely redesigning the front end and making sure that the ion optics have become much more sensitive and brighter. And also, we have taken extreme care in adding the ion optics so that this sensitivity comes also with super nice, robust platform, right? So we have seen, you would have seen in our Exceed event last week, in the presentation from Professor Matthias Mann. So he showed a lot of wonderful data from single cells, but also exciting to see that even from single nucleus, I think these are also cells where they even cut from FFPE tissues. I mean, taking single nucleus and then studying them, this is revolutionary in my opinion. And also, you get this selection based on visual observation on the cells, like it's the deep visual proteomics, or you can even have this multi-imaging based selection of the single cells which you would like to study. And And yeah, so this is exciting, and also most importantly, so this is a timsTOF SCP machine, but it's actually perfectly legal to do use this machine for other applications, like looking at high sensitivity HLA peptides, looking at cancer neoantigens, in addition to cancer biology using single cells. And also, for example, in our previous collaborations, we used to have projects where we looked at cells, and then we infect them with parasites or viruses, and then see what is the host response upon infection. And in some cases, the efficiency of infection is so low, so you really have to have this cell selected, and you might get like 20 or 30 cells. And even in this case, getting such a machine, and this machine really opens up a lot of opportunity for really experiments that were previously unthinkable. So this is truly revolutionary, in my opinion. Yeah.
Brilliant. And I also know that at the press conference, it was mentioned that you already have 4 orders in place for the new product. So what kind of labs have sort of bought the new product?
It is exciting, and it's really great to have orders already, right? Isn't it? And for cell line, and it's kind of like it is typically like, what do you say? The labs that already are doing single cells, and in terms of genomics, transcriptomics, they already have access to the single cell handling, and then they would actually run away with this kind of machine, because they can simply get also the proteomics going. And also core facilities who are experts in mass spec, and they do a variety of projects. They also have contacts, and they work with the biologists who are doing single cell work, right? So they will also make great use of the machine. So, but also eventually also it's kind of going to reach a variety of labs. And I don't think there is any single lab over time that's kind of gonna not be in a great place to use the machine. This is not going to be the case. I think this will be used by most of the labs. Yeah.
Right. It was interesting you to say that, because I know currently it's for research use only. So are there plans to bring this to the applications research market as well?
Yes, I think the starting point is the real basic cell biology research, but then there's nothing stopping it from being used in applied research, right? So this is something similar to what I said. So, for example, you can even use this in pharma research pretty soon, and also looking at personalized medicine. Maybe you have to look at individual cells and then look at all the drug like response. I think there is no restriction on what kind of research this instrument is going to be used. So I don't see any restrictions. So it's going to be a widely used machine for sure.
Right. Okay. And let's look at the other product which was launched last week, which was the timsTOF Pro 2. So how will that help further proteomics research?
Yeah, so the timsTOF Pro 2, I think I would start with timsTOF Pro, which has been a great instrument for proteomics. So whether it is cell line pull downs or studying the PTMs like phosphorylation, ubiquitination, and looking at body fluid proteomics. So either in a core facility or a cell biology lab or in a clinical research labs. So this has been a sensitive, robust, and really a fast mass spec, right? And we actually get like in-depth proteins quantifications in pretty short gradient times. And the passive technology in the timsTOF Pro platform is kind of the key to all the attributes. And actually, we even see 5,000 protein groups in about 5 minutes, which is kind of super cool. And now timsTOF Pro 2 takes all these, and now it is the successor of timsTOF Pro, and it will continue to be the kind of the workhorse for proteomics in all these kinds of different labs. And this is kind of the replacement for timsTOF Pro. But also importantly, we have to say that we have to mention this can be field upgradable. So it's the users who already have timsTOF Pro, they really don't have to buy a completely new machine to get upgraded. So this can be field upgraded, and we start doing that from the beginning of next year.
Right. Okay. So I know it offers, you were talking about, deeper proteomic coverage compared to the original timsTOF Pro. So how else has it evolved from the original machine?
Yes, it actually adds on to the existing platform. So it's the timsTOF Pro 2 has a modified TIMS cartridge at the front end, and that's coming with a Shrig design. So it adds much more robustness to the system. It has increased capacity, and also the ion packages are kind of transferred in a much more smooth manner. And then this is kind of better compared to the original instrument. So there's also, in addition, includes some modifications of the electronics. And this enables also together with other method improvements that we have. So we typically ship the machine with standard methods, and even these standard methods are kind of improved. And depending on the sample type, we can even get close to 6,000 to 7,000, or even more than 7,000 protein groups in even less than, like say, a little bit more than an hour's measurement time. So this is kind of a jump that we see compared to the timsTOF Pro. And also in low sample amounts, something in the range of 20 nanograms. And actually, 20 nanograms is sometimes not low sample amount anymore, but for 20 nanograms, I think we can get close to 4,000 protein groups in 30 minutes. So this is something, it's really, really making things faster and sensitive, right? So this is going to be the new workhorse because of all these improvements. And in addition to that, we have this run and done feature from the Pacer box, which is kind of giving you the real-time identifications. And I think this timsTOF Pro 2 with run and done feature makes it a complete package. I think it's kind of, we can say, it brings you the cutting edge proteome in more everyday life in a proteomics lab.
Brilliant. Okay, so we started off talking about what have been the developments in single cell. So let's finish by looking ahead now and talking about trends that you're seeing both in the industry, but how you predict things are going to develop going forward.
Well, trend and predicting this is a difficult question because honestly, like I think 3 months ago, I didn't think about this 5,000 protein groups in 5 minutes. Yeah. But now this is coming up, and previously 5,000 protein groups was in 90 minutes, and now it sounds like too long and too slow, right? And then so this is really going, but this is great, and I think there has been tremendous progress, and even there will be even more progress in instrumentation and data processing. And data processing has become super crucial for such analysis. And I guess machine learning and real-time data processing probably will take us somewhere like ultra low sample amounts, super fast and in-depth proteome coverage and quantification. And maybe it's not just the proteome coverage, probably it's going to be super fast in-depth epiproteome, something similar to epigenetics, where we have the proteome as well as all the PTMs quantified in a very short time from single runs. And maybe, I think it's going to be exciting, but it will be very, very interesting to see. And I think we are all part of that journey. So very excited.
Fantastic, Naga. Well, thank you very much for giving an overview of the new products. That's been really interesting. If people want to get more information, how can they find out more?
Yeah, we do have application notes, and we have the webinars and also the recordings from the Exceed event from last week, which is accessible until the end of August, I believe. And also we have lots of links available in our webpage regarding other information that is useful for the users and the customers. And also we are pretty active in LinkedIn, and we keep promoting useful content from us and our collaborators. So, yeah, but thanks a lot for your opportunity to talk with and share these developments with you. Thank you.
Fantastic, Naga. Well, look, thank you very much for sharing the information. It's been really interesting. It was interesting going to the press conference and getting more details today as well. So thank you very much for sharing the information. Viewers, if you'd like to know more about the brand new timsTOF SCP and also the new timsTOF Pro 2, then please check out the links which will be above the video, which will give you more information, not just about the products, but also the application notes and also links to webinars that Naga was mentioning, where you can sort of register. And also, obviously, check out the on-demand information webinars, which are from the Exceed event from last week as well. So there's lots of information there. And also, if you want to know about proteomics research and the different things Bruker are doing to help researchers in that area, there's loads of information there as well. You can also leave comments and questions below the video, and if you want to, message Naga directly on LinkedIn, because he is on LinkedIn, with any questions that you may have. So there's lots of sources there for more information if what you've heard today has created some interest for you. So Naga, thank you very much for sharing your time. It was lovely to see you. Good luck with the launches. I hope they go very well, and hopefully we'll catch up with you in the not too distant future.
Yep, thank you, and probably see you soon in person. Bye-bye.
Yeah, thank you very much. And thank you all for watching. As I say, check out the links above the video. Leave the questions in the comments box. Until next time, as always, stay well and stay safe. Bye-bye.